Welcome to our channel Mootechie. This presentation covers the nutritional requirements and nutritional types of microorganisms. We'll be exploring the essential nutrients all microbes need, where they get their energy, and the main classifications used to group them based on how they survive. In this slide, we dive into the core concepts of nutritional requirements and nutritional types of microorganisms. Essentially, we are looking at the essential building blocks of life and how microbes are classified based on their unique survival strategies. The main categories we'll break down are nutrient categories like macronutrients and micronutrients, energy sources including phototrophs and chemotrophs, and carbon sources which are autotrophs and heterotrophs. Understanding these categories is the foundation for the rest of our discussion. Now let's look at the common nutrient requirements for microorganisms. First, we have macroelements, often remembered by the acronym CHONPS. These are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. They form the structural backbone of all cellular components. Next, every cell needs an energy source to fuel its work. This can come from light for phototrophs or from chemical oxidation for chemotrophs. An electron source is essential for energy conservation and biosynthetic reactions. Trace metals like iron, magnesium, calcium, and potassium act as critical cofactors for enzymatic activity. And finally, water is the universal solvent, critical for transporting nutrients and facilitating all metabolic reactions. The simple diagram here shows the basic inputs and outputs for a microbial cell. Nutrients, including energy and water, go in. Through the processes of metabolism and biosynthesis, these are converted into biomass and waste products are generated. This slide focuses on the major essential ions or macronutrients in microbial nutrition. The major elements, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, make up about 95% of a cell's dry weight. They are the fundamental building blocks for synthesizing everything in the cell: proteins, nucleic acids like DNA and RNA, lipids, and carbohydrates. Cations such as potassium, magnesium, calcium, and sodium play a different but vital role. They help stabilize structures like ribosomes, cell membranes, and nucleic acids. Microbes obtain these elements from various sources: organic compounds, inorganic salts like nitrates and sulfates, or even directly from atmospheric gases. Because they are used to build the cell itself, these macronutrients are required in large amounts, often in grams per liter, to support optimal growth. Moving from the major elements, we now explore micronutrients and trace elements. These are required in only minute or trace amounts, but are just as essential. They include metals like manganese, zinc, cobalt, molybdenum, nickel, and copper. Their primary role is to serve as enzyme cofactors. These elements activate enzymes, allowing them to function. A classic example is the enzyme nitrogenase, which requires both molybdenum and iron to work. Speaking of iron, or Fe, it's particularly crucial for processes like cellular respiration and redox reactions. Microbes are so desperate for iron, they often produce special molecules called siderophores to scavenge it from their environment. It's important to maintain homeostasis with these elements. While they are essential, having too much can be toxic, so cells tightly regulate their uptake. The diagram illustrates this concept. An apoenzyme, which is inactive, becomes a functional holoenzyme when a trace element binds to it, activating the protein structure. Now let's discuss where microbes get their power from, looking at energy sources in microorganisms. The two main groups are defined by their energy source. Phototrophs, depicted on the left with the sun icon, utilize light energy. They capture this light to generate ATP and reducing power through the process of photosynthesis. On the right, we have chemotrophs. These organisms obtain their energy by oxidizing or breaking down chemical compounds. This can be organic matter or even inorganic substances. We can further classify chemotrophs based on their electron source. Lithotrophs, often called rock eaters, use reduced inorganic substances like hydrogen gas, hydrogen sulfide, or ferrous iron as their electron donors. Organotrophs, in contrast, extract electrons from organic compounds like glucose or proteins. Most disease-causing pathogens are chemoorganotrophs, meaning they get both energy and electrons from organic matter. Cyanobacteria, key oxygen producers, are an example of photolithotrophs, using light for energy and inorganic compounds for electrons. This slide introduces the other major way we classify microbes, by their carbon sources and classification. Autotrophs, meaning self-feeders, have the incredible ability to fix inorganic carbon, specifically carbon dioxide or CO2, and turn it into their own organic cell material. They use biochemical pathways like the Calvin cycle or the reductive TCA cycle to do this. Heterotrophs or other feeders cannot do this. Instead, they must assimilate preformed organic carbon that already exists, like sugars, amino acids, or lipids, to use for their own biosynthesis. Some microbes are flexible and practice mixotrophy, switching between these strategies based on what's available in their environment. The availability of different carbon sources is a major factor that shapes which microbes thrive in a community and what ecological roles they play. The graphic shows carbon coming from either an inorganic source like CO2 or an organic source with a CH3 group, funneling into the central process of carbon metabolism to create the precursor metabolites needed for biosynthesis. Let's take a closer look at the first nutritional type, autotrophs, the self-feeders. These remarkable organisms use inorganic carbon dioxide as their sole or principal carbon source. They perform carbon fixation, converting that CO2 into the organic material that makes up their cells, using pathways like the Calvin cycle or the reductive TCA cycle. We can divide autotrophs based on their energy source. Photoautotrophs harness light energy to drive carbon fixation. Examples include cyanobacteria, algae, and plants. Chemoautotrophs, on the other hand, oxidize inorganic compounds like hydrogen, sulfur, or ammonia to get the energy they need for fixation. Nitrifying bacteria are a prime example. Ecologically, autotrophs are superstar primary producers. They form the very foundation of food webs in most ecosystems by creating organic matter from inorganic sources. In contrast, the second major group is heterotrophs. Their defining feature is organic dependency. They cannot synthesize their own food from CO2 and must assimilate ready-made organic carbon from their environment, like sugars, proteins, and lipids, to use for their own biosynthesis. The most common group by far are the chemoorganoheterotrophs. This includes fungi, protozoa, and most bacteria. They derive both their carbon and their energy from organic compounds. A smaller but fascinating group are the photoheterotrophs. They can use light to generate ATP for energy, but still require organic compounds as a carbon source. They can't fix CO2. Purple non-sulfur bacteria are an example. Heterotrophs lead diverse lifestyles. They can be saprophytes, acting as decomposers that break down dead matter, parasites, which are pathogens that harm a host, or mutualists, living in beneficial partnerships with other organisms. To extract energy from complex organic substrates, they use catabolic pathways like glycolysis, the TCA cycle, or fermentation. The diagram shows a heterotrophic cell taking in glucose and amino acids and processing them through catabolism. This slide brings everything together in a comprehensive classification by energy and carbon source. The table here is a matrix that maps out the four primary nutritional types based on these two criteria. In the top left quadrant, we have photoautotrophs. They use light for energy and CO2 as their carbon source. They are the primary producers, with examples like cyanobacteria and algae. Top right are the photoheterotrophs. They use light for energy but require organic compounds for carbon. Examples include purple and green non-sulfur bacteria. The bottom left quadrant shows chemoautotrophs. They oxidize inorganic compounds for energy and fix CO2 for carbon. Nitrosomonas and sulfur-oxidizing bacteria like thiobacillus fit here. Finally, the most common type, found in the bottom right, are chemoheterotrophs. They oxidize organic compounds to obtain both energy and carbon. This group encompasses most bacteria, all fungi, protozoa, and even animals like humans. The remaining values in the table follow the same logical pattern, clearly showing how energy source and carbon source combine to define a microbe's lifestyle. You might be wondering why nutritional classification matters. This slide highlights four key areas where this knowledge is applied. First, in cultivation and media design. To grow a specific microbe in the lab, you need to design a selective media that supplies its exact required carbon and energy sources. Second, it provides ecological insight. Knowing a microbe's nutritional type helps predict its role in biogeochemical cycles, like whether it's fixing nitrogen or decomposing carbon in an ecosystem. Third, there's clinical relevance. A pathogen's metabolic profile can help identify it and even predict what virulence factors it might use within a host environment. Finally, it's crucial for biotech applications. Optimizing industrial processes, whether it's fermentation for food production, creating biofuels, or using microbes for bioremediation, relies on understanding and catering to their nutritional needs. The icons represent these interconnected worlds: the lab flask, the clinical cross, the factory for industry, and the leaf for ecology, all connected through the central concept of nutritional types.
Let's recap with a summary and key takeaways. Microbial growth fundamentally depends on macronutrients like carbon, nitrogen, and phosphorus for structure, and micronutrients like iron and zinc for enzyme function. We classify microbes based on three primary sources: their energy source, light or chemical; their carbon source, CO2 or organic; and their electron source, litho for inorganic or organo for organic. In nature, autotrophs play the critical role of primary producers, building organic matter from scratch. Heterotrophs drive the decomposition and recycling of that organic material. Understanding these nutritional types isn't just academic, it directly informs practical strategies. It guides how we design culture media in the lab, analyze ecological interactions, and develop industrial bioprocesses. The web of terms around the central microbial metabolism illustrates how these concepts are all interconnected. Thank you for exploring the fascinating microbial world with us. We hope this breakdown of nutritional requirements and types has given you a clearer picture of the invisible engines that drive so much of life on Earth. Remember, these fundamental concepts are the key to unlocking everything from diagnosing an infection to harnessing microbes for a cleaner planet.